Okay, in our previous video, we were talking about how cells can regulate protein synthesis. We can turn off a gene or turn on a gene and manipulate gene expression by making sure RNA polymerase is blocked or by making sure RNA polymerase does come and transcribe a gene. Well, what I want to turn to is what happens when something goes wrong. And this has to do with mutations. And so when we're talking about mutations, we have to kind of contend with a lot of misinformation that's out there between X-Men and Spider-Man and all sorts of superhero cliches of mutations giving superpowers, or horror movies, monster movies that talk about mutations. We definitely get some really strange ideas out there. A mutation is nothing more than something that is changing the DNA sequence. Now, most of them can happen in areas that aren't coding for anything, in which case we don't really notice these mutations. Other mutations do occur in coding sequences like proteins. Sometimes these are silent mutations, meaning that they don't have any change. So let's talk about why that is. So mutations are when the DNA or mRNA sequence is changed. This happens most likely in DNA replication. There are some mistakes that can happen during transcription. And so let's take a look at some of these types. Now, it says wild type up here, but wild type simply means that this is the normal DNA sequence. Now, notice, and this is what you're doing in the Monster Mystery Lab, you're taking your DNA and making mRNA, and then taking mRNA and finding your amino acid sequences from it. And so notice we have TAC, AAC, GTC, ACC, ATT. Now, in the first mutation, notice we have, instead of AAC, we have AAT. Now, that's going to change our mRNA strand to UUG to UUA. Now, this is called a point mutation because only one base changes. We went from AAC to AAT. That's 1 point in the DNA. Now, that does change the mRNA strand as well, 1 point. Now, in this case, though, UUG and UUA both code for a leucine amino acid. So in this example, this is a silent mutation. Nothing changed in the actual amino acid sequence. So we call that a silent mutation. No change in the amino acid. Now, let's take a look at this one in the middle, a missense mutation. In a missense mutation, in this example, we changed it from AAC to AAG. Now, in this case, this does change the amino acid. We go from UUG to UUC. And if we go to our codon chart, that means we went from leucine to phenylalanine. Now, you don't have to memorize these. The idea is that you are using the chart to notice that these amino acids are different in this case. So instead of leucine, we now have phenylalanine. And we call this a missense, like a mistake. And we changed one amino acid. Now, a missense just affects one amino acid. This may or may not affect the actual protein. We'll talk about that in a bit. Now, the last type of point mutation is when you change it to a stop codon. And that's going to be really bad because if it stops at the beginning, that means you go from an mRNA that would make 300 amino acids, and you could potentially stop it at 10, and you suddenly lost the bulk of your protein. Now, we call this a nonsense mutation. And what I like to think about is, well, we just made a nonsense protein. We just stopped it and we've not made it. And you can imagine that's a really disastrous mutation. Now, the other type of mutation is when we insert or delete a base. Now, notice we go from, in our top, we had TAC. Down here, notice we have TAT, but we didn't change it. What we did is we added. So it's not that we changed the TAC, it's we added a T to it. So notice there's a T between the A and the C in this last example. Now, that's going to be really bad because notice if we do that, look at what we just did. Every codon suddenly changes. Remember, a codon is every 3 letters, and that's how we make our amino acid strand. So up here, we decided that our codon is AUG. That's our 3 letters. That's how we found the amino acid methionine. And then we went UUG. That's how we found our amino acid leucine. But if we insert a base here, notice that changes everything down the line. Instead of AUG, we suddenly have AUA. And instead of, what's our second one, UUG, we suddenly have GUU. And notice what that does to the whole amino acid strand. That makes an entirely new amino acid strand after the insertion. And we call it a frame shift because we shift the reading frame. And I'll give you a good example on the next page. So here's just another example of a frame shift where we change the reading frame because we've inserted or deleted. So notice we add one, and that changes the rest of our amino acids. And this is called a frame shift, an insertion or deletion. Now, these could also be silent if you had a big chain of like A. Shifting that may not change that much. But it also could be nonsense because it could stop. You could introduce a stop codon in there, and then everything stops. It could theoretically be a missense if it's at the very end. But generally speaking, a frame shift is going to change a lot of amino acids. One way to do this, one way to think about this would be if we make a very simple sentence like, the fat cat ate the rat. Now notice these are all 3-letter words. That's going to help us understand how we change it. If we only change the F to an H, we now have made the fat cat ate the rat to the hat cat ate the rat. And that would be a point mutation. We have changed one word because of that. Now, if we delete one letter, notice how that changed the rest of the sentence. Or if we added a base or letter, notice that changed the rest of our sentence. So a deletion or an insertion or duplication is going to change things. So let's go over an actual example of how this happens, and then you as a class are going to watch this TED Ed video. But let me go ahead and show you. In our red blood cell, I told you it was the tale of 2 cells. Well, one is the normal cell. The other one would be a sickle cell. And a sickle cell red blood cell has a mutation in just one spot. It is a point mutation. But this ends up being really important because if you change the DNA sequence where it changes one amino acid, so let's take a look at this. Here's the DNA, and on this codon CTC, it's changed to ACAC, and that changes the amino acid from glutamate or glutamic acid to valine. That's the mutant. Now, that is enough to cause a really big problem. And let's take a look at our chain of events. What ends up happening is normal hemoglobin kind of looks like a little basket or a wreath. And along this outer wreath, you can hold your oxygen molecules. And that would be normal hemoglobin. But in sickle cell hemoglobin, in low oxygen conditions, what ends up happening is hemoglobin clumps together. And it's that clumping that causes a disaster. So in normal red blood cells, they kind of have this jelly donut kind of shape. But in sickle cell red blood cells, if the hemoglobin starts chaining together like this, it causes the cell to buckle and warp. And that becomes a problem, as you'll see in the video. Now, here's why this can occur. And follow the logic, follow the step by step here. If we change the DNA, you're going to, by our rules of transcription, you're going to change the RNA. If we change the RNA, we could change one or more amino acids, as we just went over. If we change one amino acid to another, we could change, and this is the key here, we could change whether or not we have a positive charge on one amino acid. We swap it for an amino acid that has a negative charge, or we may swap a negatively charged amino acid to a neutral amino acid, or vice versa. So this is kind of the trick. If you change the amino acid, you could change the charges that cause the protein to fold in a proper way. And as we learned way back when with the enzymes and other proteins, it's the shape of that protein, that lovely globular shape of that protein. And I have it right here. Here is that shape of my molecule. Here's my amino acid strand. And if this is my hemoglobin, one change of an amino acid could mean that instead of these guys being joined together, they fall apart. And that affects my shape of my protein. And that becomes very, very important. All right, so hopefully that helps us understand why. You're going to do an activity together in class, and then you will have some time to work on your monster mystery lab.